My talk is designed to give you a general overview of genome-wide association studies and of heritability estimates that we can obtain from different sources. It then follows through to a little bit about how my group has contributed towards those efforts to better understand the genetic basis of common complex disease. And it finishes almost with a computer science talk about how we can estimate regression coefficients in very large-scale data sets better. So this is going to be one of those talks where nothing works, isn't it? Yep. Okay, so anyway, I can use the thing, it's fine. Okay, so I'm going to start by defining what I mean by a common complex disease. So something that, that affects 1% or more of the human population, and this could be anything from stroke to cardiovascular disease to type 2 diabetes to schizophrenia. And current evidence from genome-wide association studies suggests that many tens, hundreds, if not thousands of independent genomic regions contribute towards risk, combined with many multiple non-genetic factors. These can be environmental risk factors or just stochastic chance events. And a simple statement that as you get many factors, things become normally distributed. So as you move to one locus, individuals carry 0, 1, or 2 risk alleles. And as you move to 4 loci, it starts to move towards looking like more of a normal distribution. So this leads us to, there's a wealth of theory in quantitative genetics about the liability threshold model. And so if we assume there's a phenotypic liability of risk, and there's a threshold point at which individuals in red here start to display symptoms that if they went into a doctor's surgery, they would be classified as having a particular disease. And all we have to assume here is normality. So we have to, which is appropriate for anything that's multifactorial. It doesn't have to be highly polygenic or infinitesimal. It can just be a few genes. And the key is that we have one unimodal distribution to describe our risk. And most of the effects are governed by the proportion of the population that are diseased, which then give you the threshold points, the density, and the mean phenotypic liability of the diseased group. Now, obviously, I came from Edinburgh, and there's a great history of quantitative genetics research in Edinburgh. This is a brief and incomplete history of the liability threshold model that I don't expect you to write down. But it begins with Pearson in the 1900s and moves all the way through. And I'm going to talk a little bit about the Falconer work in 1965. And I'm speaking a little bit about this because I think it's largely overlooked. And there's a lot of parallels between predicting a response to selection, where you have a first generation that's not selected and selected, and then in the second generation, you take the selected individuals and you have their distribution and you select on them again. For the quantitative genetics of disease, it's a similar kind of process where we have the population of not diseased and diseased, and we take the diseased individuals and we look at their relatives, and we look at individuals that are not diseased and diseased. And we have the same kind of theory that basically predicts what's the change in this threshold point across relatives, and given the difference in the threshold points from the population to the relatives and the relationships between the diseased individuals, one can infer the heritability. And in Falconer, 1965, provided a graph just to do this. So if we take the relative risk to relatives, the proportion that are affected given that they have a relative that's affected, divided by the proportion that's affected in the population, so the population prevalence here. If we take the baseline schizophrenia risk to be just under 1%, this is a hard enough thing to estimate in a population itself because it depends on how you diagnose things. If we take the risk to relatives, the risk to siblings to be 8.6, we can calculate K as 8.6 times 0.85 to give us a 7.13%. And we can move from the incidence in the general population, which is just under 1% here, up to the incidence in relatives, which is 7.31, to get an estimate of this line of the heritability of schizophrenia, which puts us at an estimate of about 70%, just based on family data. And this is supported by other literature, where if we look at the risk factors for something like schizophrenia, the biggest risk factor is family history. And this is why when you walk into a doctor's surgery, the first thing they ask you is, what's your history of X, Y, and Z? And that implies that basically genetics are important. And there's also evidence of a genetic contribution of the risk to relatives for anything from major depression, ADHD, schizophrenia, bipolar, or autism disease, where the prevalence is much higher in first-degree relatives with an affected individual than the baseline population. And so we have a process for common complex disorders where, unlike Mendelian disorders, there's no clear classification, right? So unlike Mendelian, there's no clear pattern of inheritance that you can follow. They tend to run in families. But we actually, in humans, have very few large-scale pedigree studies. Unfortunately, well, not unfortunately, we live for too long. So it's hard to collect data with any depth, and it's hard to collect data on enough relatives of those that have a disease. And actually, most people, if you have a liability threshold model, they don't have any family history at all. So if you have two parents that sit just below the threshold, they're quite likely to have a child that sits over the threshold, but that individual will have no family history of the disorder. So the overarching aim of what I want to do in my career is to better understand the genetic basis of disease. And by doing that, all I hope to do is to provide a better measure of family history that we can use in the clinic. That's the overall game. I wish this worked. So how useful are heritability estimates from family data? Well, this is a study that we published in 2017 in Nature Genetics, where we took the phenotypic correlation estimated for a series of family members from 172,000 18-year-old brother pairs from records in Sweden. And this is conscription records, so 18-year-old siblings will go into the army, they'll have lots of things measured about them, and we can calculate the phenotypic correlation from monozygotic twins, dizygotic twins, siblings raised apart, half-siblings and half-siblings raised apart. And from these simple correlation estimates, we can estimate the heritability, the proportion of phenotypic variance explained by genetic effects. And we can do that in two ways. One, in the dark colors here, by accounting for the assortative mating estimates that we made in this 2017 paper. So humans are interesting because they mate assortatively for almost anything. So you and your partner, you and your heterosexual partner, if you are heterosexual, if you have a child, you will tend to phenotypically resemble your partner on average in the population for height, body mass index, educational attainment, schizophrenia risk, anything like that. And this positive assortment creates positive assortment at trait-associated loci, and it influences the heritability. And so if you account for that, what you get is stability in the height heritability estimate, irrespective of the relationship group that you use. But for body mass index, the estimate of heritability that you get from monozygotic twins is nearly the same as height, but then it decreases as you go to half-siblings raised apart, implying simply that obviously relatives don't just share genes, they share a common environment also. And that's one way of breaking down that common environment sharing to get a more accurate estimate of what the true heritability is. With the advent of marker data, so we have lots of individuals measured at lots of SNP markers, we can actually repeat this experiment by, rather than using what their expected relationship is from a pedigree, we can use their realized relatedness estimated from genetic marker data. And there's various equations to do this, but you can generalize the case where basically all we're doing is we're taking our markers, 0, 1s, and 2s for different genotypes, we're subtracting by the mean, 2 times p, the allele frequency, and we're essentially just estimating a covariance matrix between all of our individuals. And we're weighting this in some degree, depending upon the allele frequency raised to some power s. And the s parameter that you use depends upon your a priori belief of how important rare variants are, and there's a paper that discusses that here. And then we're using a model where we have y, x is all of our SNP markers, and b are all of their effects, and this is a residual vector here. We can represent this model in exactly the same way with w and u, where u are the genetic, the breeding values in the animal breeding literature, or the genetic values in the human medical literature, and which we can estimate using this genetic relationship matrix. So we can partition the variance in y into 2 components, one attributed to resemblance between relatives and the other that's residual. And this leaves us in the field. This is an example for human height, and we have different variance estimates depending upon how we do the study. So in the bottom here, in this purple color, we have 16% of the variance explained by genome-wide significant SNPs. So if I identify markers and go into an independent sample and make a prediction, as I'll explain in a minute, we explain 16% of the variance. If we use just a standard SNP chip, we get 45% of the variance explained. If we get imputed sequence variants, we get 60% of the variants explained. If we get whole genome sequence data, as I think Peter Vischer will explain to you next week, you'll get close to this 80% estimate that you get from family studies. And these differences from 16% up to 45 is a result of sample size and power, as I'll explain in a minute. This is a result of better tagging of on-genotype variants, and this could either be because there's rare variants that contribute towards the phenotypic variation that we're not capturing even with imputation, or that our family estimates are upwardly inflated, as I showed for BMI. So how can we make this gap here? So this is our prediction accuracy that we get from GWAS. How do we push this up towards this heritability, and how high can we possibly go? The pink one here at the bottom is the variance explained by genome-wide significant SNPs. So if I identify markers in an association study, and I take single SNP regression, so just using single SNP regression, I'll talk about other ways to do that better, and that's the focus at the end of this talk. So here's an example for educational attainment, and a GWAS in 2013 of 127,000 people that identified 3 GWAS loci. If we move forwards to 2016, to a sample size of 294,000, we get up to 74 loci. And a more recent one that we were involved in 2018, you go up to 1.1 million people, and you get 1,271 loci, simply as a result of your shrinking standard errors as your sample size increases. If you take these results, then what you can do is you can take a training set of information that you've used to estimate these effect sizes. You take these effect sizes into a testing set of data where you have genotypes of the other individuals. You create a predictor and you assess how good your prediction accuracy is or how well it does. And theory tells us the expectation of the R-squared between the phenotype and your predictor of the phenotype is governed ultimately by the heritability, the variance explained by the covariant, by all of the markers, by all of the covariate effects. But it's also scaled by 1 plus m, the number of causal covariates, to the ratio of n over h squared. And this says, so the variance explained by the covariates, the heritability really sets the upper bound. And this makes sense. Obviously, we can't predict something from DNA more than what DNA explains. But also, the ratio of the causal covariates to the sample size is important, i.e., how well we estimate the effects matters. So because we don't estimate the effects perfectly, our estimates are the truth plus some error. What it means is that we can never, ever approach the true heritability. We'll always be under it. And how much we're under it depends on the methods that we use. And at the moment, from single marker regression, when we go for height, body mass index, schizophrenia, bipolar disorder, and Alzheimer's disease, we have quite variable prediction accuracy at the moment across these traits, where for height we're sitting below 20%, body mass index somewhere below 10%, schizophrenia around 5%, bipolar around 3%. Alzheimer's disease we're doing quite well, and that's because there are variants segregating in the human population that have quite strong effects. So that's my little whirlwind tour of where we are right now in the field. And the rest of this talk focuses on, can we do better, given the data that we have? Yep. We are taking estimates. These values come from, we are estimating them in one sample, going into an independent sample and making the prediction. So it's not any cross-validation or leave one out, leave a sample out. It's going into an independent study. I'm assuming that the underlying effects are the same and that the LD patterns are the same, which is obviously not strictly true. What we've done in most of this is gone from one big European sample into another European sample. FST in Europeans is not that big, so you don't lose that much. None of this talk is about cross-population genetic prediction. Okay, so can we do better given the data that we have? Well, what data do we have? Now is an amazing time to be a quantitative geneticist because we have lots of data, right? I have more data that I can manage. We have the UK biobank study, 500,000 people from the UK. MOBA study, 100,000 mothers and their children. We have the Estonian Genome Center, which at the moment is 50,000 people, going up to 5% of the Estonian population. We have the US having 2 major initiatives to recruit over 1 million people. And this combines medical health records, thousands of phenotypic records on these individuals tracked for the rest of their life after they enroll in the study, combined with eventually whole genome sequence data. And This is an extreme situation where actually n, our sample size, is much, much less than m, the number of markers, right? So we're in a standard regression framework where n is much, much smaller than p. Okay? So the approach to this in genome-wide association studies, as said by a member of the audience, is single marker regression. So we have one column here of marker values. We regress that onto the outcome. We obtain one estimate. We throw that away, and we do the next one, and we throw it away, and we do the next one. And there's clear problems with this. It doesn't account for the LD in the genome. It has poor power for discovery because of the properties of the way in which we do the estimation. It has poor prediction as a result of that, and obviously we have to imply quite a high multiple testing threshold in order to deal with the associations. What we would ideally like to do is to model all covariates jointly, right, in this scenario. And one way to do this is through penalized regression approaches, such as lasso or ridge regression, where we're applying a shrinkage parameter onto our covariates. But this typically, its implementation relies on cross-validation, and a lot of its application is quite inefficient in the way in which it's coded. So there's a whole field in genome-wide association studies of doing the analysis one marker at a time, and then moving to an independent sample, calculating the LD, and trying to put it back in. Right? So trying to formulate an approximation for a ridge regression or for a lasso. Right? So to try and take estimates that don't account for LD, but to put it back in. Right? I am one of these people, and so we had a paper in Nature Communications last year that was about improving genetic prediction by leveraging correlations across disorders. There was a paper in 2015 in Nature Genetics that came up with an approach where you can take summary data and take LD patterns and exploit single marker regression to tell you what the genetic correlation is among different disorders. So you see some interesting patterns, and one of the main things is that we haven't been able to estimate this before, really. We haven't got large-scale pedigrees where we estimate lots of these things together. We don't really know what the genetic correlations are in the human population. And you get some things that make sense, so high positive genetic correlations between schizophrenia, bipolar disorder, depression, high correlations between morphometric measures, et cetera, et cetera. And if I wanted to predict something like type 2 diabetes, which is on this list, can I use these correlations to help me to improve its prediction? So I know type 2 diabetes is highly correlated with BMI and childhood obesity, and I don't have many observations of type 2 diabetes, so I have low power. But what I do have is hundreds and hundreds of thousands of estimates from body mass index. So can I borrow that information to help me predict type 2 diabetes risk? And that's what we did. Yep. In this here? In this study here. This study here regresses essentially the test statistic onto the LD. And because of single marker regression, you have a higher chi-squared value, a higher test statistic value for markers that are in LD with more stuff that tag more variation. There's a relationship between effect size. And so you do that regression, and the slope of that line, if you rearrange the terms, tells you something about the heritability. And if you do that, taking the product of the z-scores from the estimation, it can tell you something about the genetic correlation. That's how that works. And so that just provides an estimate of the genetic correlation. Yeah.
I mean, how is it related to the phenotypic correlation? So are they also strongly correlated in phenotypes and have a strong genetic correlation?
That's very hard to estimate for something like schizophrenia, bipolar, and depression, because do people have all those things at the same time? For something simpler, yes, there's a correlation between your birth weight and your birth length and your eventual height, yes. Most of the time, I'm going to say this tentatively, I can think of very few examples where we have phenotypic correlation partitioned into genetic correlation and residual correlation, where those two things are not aligned. Right? I can't really think of an example right now where one is positive and one is negative to give a zero. But it's possible, yes. Any other questions? I like interruptions, it's good. Please interrupt me as we go along if anything doesn't make any sense. So what we did is we take summary statistics for a given trait, and we take summary statistics from another trait. And we have an S-BLUP, so best linear unbiased prediction approach, where it's an approximation of a Ridge regression. So we're taking these single marker least squares effects, and we're turning them into an approximation of what you'd get if you'd have done the Ridge regression, okay? Just by putting the LD back in and shrinking them according to the heritability estimates that we obtain from the previous approach that I discussed here. And yes, so in a Ridge regression, as I understand it, in theory, it's the error variance divided by the effect size variance. That's the theoretical parameter for the lambda value. And so if we know the heritability and we guess at the effective number of markers in the genome at 50,000, then we get an estimate of the variance B, and we just assume that the error variance is 1 minus the heritability. We can also do some form of cross-validation where we work out what it is that gives us the best R squared in another population, and there are other approaches that do that. I don't like that so much as using what the theory is, but that's up to you. Anyway, so we get these estimates, and then we can then approximate what you would get if you did a multivariate REML model. So if you did a bivariate approach here, just by knowing what the genetic correlation is between the two traits, the heritability estimate, and we can weight them. And in the animal breeding literature, this is called a selection index, where they take two traits and they weight them together to predict what the selection response would be, solely dependent on the RG and the H squared. So there's lots of approximations here, obviously, right? But it seems to work relatively well. So we have for bipolar disorder and schizophrenia, we have the correlation between the predictor made from GWAS estimates, so simple least squares estimates, what we get from BLUP, just putting the LD back in, as I said, approximating a Ridge regression, and what we get from multi-trait BLUP if we borrow information from schizophrenia to help us to predict bipolar disorder. The other way around, we're predicting schizophrenia, borrowing information from bipolar disorder. Okay, so we get the improvements in prediction that we would kind of expect under the theory for a mixed linear model. And for predicting type 2 diabetes, we can see diabetes here, the correlations are very small. BMI actually helps us very much to predict type 2 diabetes, as do lots of these other traits. And when we weight them all together, we get this gold-colored bar. And this is still a tiny amount, but this actually represents a 42% increase above that bar. And so in theory, as the sample sizes grow and the prediction accuracies increase, we will always maintain this gap. It will just grow like this together. So that's one way of doing this, and it shows that jointly fitting the markers, even approximately, can help improve prediction accuracy, and that borrowing information from genetically correlated traits also helps. But that's an approximation. And that works okay in the situations where we're never going to have access to the individual level data, which is some of the time. But what we'd actually like to do is do this at the individual level. So we'd like to be able to run this model when we have individual level data, or I would, and I hope that by the end of this talk you share my enthusiasm. So the idea is to model covariates jointly to obtain partial regression coefficient estimates, to properly account for the LD, to improve our power, to do variable selection without having to do lots of multiple testing, and to describe the genetic architecture behind traits. So I'll discuss that in a minute. And the idea basically is that we want to sample the marker effects iteratively, conditional on the data, right? So we want beta to come with the probability of beta 1, given the data that we've observed, X, the genotypes, and Y, the phenotypes, and all the rest of all parameters in the model that we've estimated. That's what I'm looking for. So the animal breeding literature helps me again, because I've spent my whole career stealing from it, there's a model formulation called Bayes R. And basically, it's a simple regression model where Y, you have an intercept term, some markers, their effect sizes, plus some residual error. The log likelihood of this model can be described just using the normal distribution. And we assume that the beta distributions, we're assuming a Dirac spike and slab model where we have a scale mixture of normals. So we're assuming that there's a point mass here at 0 and a probability pi 0 that the markers have no effect, right? So a spike on 0 where markers have no effect. And then markers can come from a series of normal distributions and their variance with different variances. And we set these variances depending upon the total variance explained by the genetic markers in total. So the sum of the beta squared. So we assume that these are standardized and that thus to a z-score, variance of 1, and thus the sum of the squares of these terms here gives us the amount of variation explained in y. And we use that to sample what we call sigma g, the total variance explained by the marker effects. And we use a series of constants to basically break this up across these mixture groups, where we have a small distribution of effects, a bigger distribution of effects, and an even bigger distribution of effects. So we're assuming that some marker effects are 0, some are small, some are bigger, and some are even bigger. And we're approximating whatever the distribution of true marker effects is using this mixture of normals. Exactly, yes. And I'm not using a t-distribution or anything like that. I'm just using a series of normals, but also different from the first implementation of Bayes-R, I am not setting these variance parameters, I'm estimating them from the data. So we derived the updates for this. We use a coordinate updating scheme, so a random scan Gibbs sampler, essentially. We take a marker at random, and we want to estimate what its inclusion probability is, and given that, we'll determine what distribution it falls from. Given that, we want to sample its effect. That's the idea. This is useful when going up to scale that we have. So in the UK Biobank, we have 500,000 individuals. We're trying to do this estimation on 14.5 million markers. If we sample all the inclusion probabilities first, as has been proposed in the literature, use the markers in the model, and then just do the multiple regression equation, we're still inverting a matrix that's huge, right? So we still have high memory requirements. It's still taking a long time. So instead, we return to old ways of coordinate updating, which are known to converge slowly, geometrically. But basically, given the sparsity of the problem that we have and the prior that we have, we assume all the effects are independent, right? So it's like an independence prior. And the sparsity of the problem, we actually achieve convergence quite quickly, because quite quickly the coefficient matrix that we have has an eigen value that's quite small, so it converges quite quickly. If I'm not providing any more details than that. Right, so coordinate updating, the full conditional posterior is a product of the likelihood I just showed and the priors, and that reduces down to this term, which gives the Gibbs sampling update that our markers come from a normal distribution with a mean that's given by x transpose y, so the dot product, divided by this sigma jl, which is x transpose x plus some shrinkage term here, which depends on the area variance divided by variance L, and variance L is the variance contributed by the mixture group. Okay. Having what we want to do first, though, is sorry, one step first. What we want to do, and the nice thing about this is that we derive that actually what you're estimating here, if you have two markers, x1 and x2, that are correlated with each other, and you're trying to estimate the second effect, and x1 is already in the model, then the estimate that you get for beta 2 is basically a shrinkage estimate of the partial regression coefficient. So you have the correlation here between y standardized for everything else and x2 minus 1 over n times the dot product of x1, the other covariate, times yc, divided by 1 minus the correlation between the two markers. So as this approaches 1, this correlation approaches 1, this approaches 0. If these effect sizes are the same, then basically you will never include a correlated marker in the model. So it induces this, if you have LD and you have blocks of LD and there's a marker here and you pick this one, nothing else in high LD will enter the model unless it really has an independent effect. That's basically the summary. Before this, we have a Gibbs sampling scheme, so what we want to do is actually estimate the inclusion probabilities. What we want to do is first select the mixture group that we're sampling from, and the probability of inclusion, so given the mixture group 0 versus mixture group 1, for example, we derived as given this equation, and basically it's just the square of the estimate of the regression coefficient divided by what's this n minus 1 is x transpose x plus this shrinkage term here, given the variance component, times the error variance. And so the stronger the effect, the more likely it is to enter the model. And this gives us a simple baseline algorithm that I'll talk about how we can adapt, where we have so at time t minus 1, we have our old estimate, and we take the marker value, we times it by its old estimate, and we add it to the residual that we have at the moment. We then calculate the cross product between the marker and that new residual that we've created, and divide it by this sigma term to give us what the beta estimate is, to go into the updating equation to determine the inclusion probability. So that goes into here, over there, to determine the probability of inclusion. Then, if it's included in the model, what we want to do is sample the new value of beta j. If it was never in the model before, and it's 0, then you're adding nothing to the residual, and so nothing changes. And then if you sample it, it's 0, so nothing changes to the residual. If it is in the model, and you do sample it, then you have a difference between what it was and what it now is, multiplied by the marker value, and that gives you your change in the residual from your estimation. So it's a residual updating, or in the machine learning literature, a backfitting algorithm. And the way in which we've set it up is very nice because it facilitates us to split the information, so to parallelize the Gibbs sampler, as I'll explain in a minute. So it allows coordinate updating, and we can pass this information, as I'll talk about in a minute. But the general take-home message, if none of that made any sense, is that we sample the parameters, the SNP effect sizes, thousands of times, tens of thousands of times, to get their distributions. The proportion of non-zero betas throughout the iterations gives us the probability that a marker has an effect. The probability is included in the model. And assigning the markers to different distributions describes the distribution of the effect sizes or the polygenicity of a trait. And I'll give you a demonstration of that in a minute. As an aside, we applied this baseline model to DNA methylation data. We have a paper on bioRxiv about this, where we have a 450K array that tags methylation marks in the human genome. And there's a wide variety of methylome-wide association studies, and they borrow from genome-wide association studies and look at the association between a disease or an outcome, BMI, smoking, age at death, all these things, regressed onto each methylation probe one at a time. And methylation data is fundamentally different to genome-wide association data in that you have a very heterogeneous sample of different cell types depending upon the tissue that you take it from. And so there's a huge variety of literature about calculating principal components of the methylation data, fitting sparse principal components, fitting latent factors, et cetera, et cetera. And what we wanted to show mostly about in this paper is that if you're fitting everything together, right, if you're fitting all the methylation probes at the same time, you're controlling for the background when you're estimating any one marker effect. Okay? So you don't have to bother fitting all the PCs if your aim is prediction. You don't have to worry about the structure of your data so much. And also, we extended the model to to enable us to say, okay, let's say we have BMI. We want to fit some fixed effects. Fixed effects, we still sample them. They're still random, of age and sex. We want to divide our variance component up into two. So we're essentially fitting a hierarchical model where we have two variance components now. One for the variance explained by the CpG or methylation sites, and another explained by the SNP markers. And so that's nice because we estimate these CpG effects conditional on the SNP markers, and we estimate the SNP markers conditional on the CpG effects, and estimate the CpG effects all conditionally on each other. So we get a real estimate of how we can partition this variance into two groups. We double counted them, yeah, and let the data tell us what happens. And I'll talk about why in a minute, because I think the methylation changes that are associated with the trait are a consequence rather than a cause. So I don't think it really matters when you do that. We tested our approach against GLMNet, Lasso and Ridge, latent factor mix modeling, again, Lasso and Ridge, according to different amounts of correlation between our effects and the background. So we take the first PC of all the methylation data and created a correlation between our genetic effects or our methylation effects and that latent structure. And what you can see is that we basically control well for it. So so this is the correlation between our estimates and the truth, the mean square error, the correlation between what we estimate, what the background structure is, and the genetic values. It's not overinflated here, and we get accurate estimates of what we simulated the variance explained by the CpG sites to be. And when we apply this to BMI, what we see is that environment, so residual variance is around 10%, genetic markers explain just over 20%, which is what you'd expect, and all the rest is explained by methylation probe data. So if I take a blood sample from you and I look at something else, not BMI, but type 2 diabetes or cardiovascular disease, then I'm going to capture, so I don't know your actual disease status, but let's say you're on your way to getting cardiovascular disease. If methylation variation explains quite a lot of the variation in the trait, then I'm able to capture that process that you go through, because methylation changes in your DNA are a consequence of your environment that are getting you towards that disease state. Not only that, but we can learn things about the process, and this looks horrible, and you won't be able to see anything, but basically this is plasma lipoprotein and particle levels, cholesterol transport, sterol metabolism, anything from feeding behavior, right? All different pathways involved in obesity and BMI. So you can take the posterior distributions and do an enrichment test on them, and this is what you obtain. So you can learn about the pathways also that underlie your progression. Not only that, but we beat Lasso in terms of prediction in an independent set. So we go into another Scottish data set. That's the R-squared from Lasso, and we do a little bit better depending upon whether we want to predict BMI or smoking. So this just demonstrates that a blood sample can actually provide a large amount of information about you as to where you are on a path to disease. It's essentially regressing the phenotype onto itself, so we explain a lot of variation. And by doing that, we can also learn a lot about the underlying biological pathways involved in that process. But that model looks great. Well, I'm happy with it anyway. But does it scale to the data sizes that we have? So this is the lengthy process that we went through over the past year in my group, which is to try and handle data on 500,000 people and over 10 million genetic markers with low RAM use. And we started by using Intel's TBB library. So this is a brief overview of some computer tricks that we've used. We can actually pre-process the data to a sparse format, and I'll discuss that in a minute, using the properties of genetic data. We can then compress the data, we can memory map the file, and then we can have a series of nodes that are decompressing and feeding the data to the algorithm as it needs it. So what this means is we can simply avoid the step of taking the data, loading it into RAM, and then having it there. So what this means is that you could run an analysis, it would take a very long time, on your iPhone, on 500,000 people. Okay? So we can use the memory quite well. Then what we can do is we can use bulk synchronous Gibbs sampling. So we can, because of the sparsity of our problem, right, for most traits we have 14.5 million markers, and only 30 to 70,000 of those are in the model at any one time. The rest are zero, or assigned to the zero mixture group. So if I break up my algorithm into sections, and I take a marker here, a marker here, a marker here, and a marker here, and I have 300 groups, probabilistically, only 1 to 2 of those are actually updating an effect at any one time. Most are zero and have no effect, and nothing is changing. So all I need to do is sample a marker, right? Take 300 markers, sample each one. When I've updated something, I need to pass my residual vector, my epsilon vector, or the change in that epsilon vector, to all of those 300 workers, and then they can all take another column. That's the idea. So we're just parallelizing that in a way that still maintains a single Gibbs sampling chain. Okay. So it's not parallel, it's still synchronous, but it's kind of blocking things in bulk. Right? And you can do this basically within one node. So you can utilize all of the CPU, all of the threads on one node, and that's great. But can we do better? Yes. So we can A, utilize the sparse information as I'll describe, but B, break everything up across nodes. So the main thing for us is that dot product between the marker and the phenotype, because we're doing it 14.5 million times, and then we're doing that 10,000 times. So it's quite a high computational burden, and the length of that vector is 460,000. So what we can do is we can do, when you take a dot product between the residuals and the markers, for zeros you do nothing. For ones, you just sum them up. And for 2 values, you sum them up and double them. Right? So rather than doing a vectorized dot product, what we can do is a series of sums. And that saves you 200% of time, which is crazy, right? But it does. And then we can split the data up across nodes. So we can allocate 300,000 markers to each of a series of nodes. We can sample the parameters in parallel and synchronize it again across nodes and within nodes, as I showed you. So by doing that, we can have a stable Gibbs sampling algorithm that enables us to do one iteration in 100 seconds, which is still not fast enough for me, but we're getting quicker all the time. And I think this opens up the way for a wide range of penalized regression approaches to be applied to large-scale data sets on the actual individual level data, rather than trying to approximate it in some way. We can show that true positives recovered against false discovery rate, that for our approach in green, we basically have more power and a better true positive rate given the false discovery rate than anything else currently used in the field. If we do a simulation study, very simple one, where we break up a number of populations, that's how they would look in a PCA plot, and we assign different genetic covariants with ancestry. So if we say, okay, well, our genetic values are not at all correlated with ancestry, or our genetic values are very highly correlated with ancestry across populations, there's genetic differentiation across populations, does it influence our estimation error, or does it influence our false discovery rate if we set a posterior inclusion probability threshold of 95%? And basically, we're kind of well controlled. The only issue where we fall down, which is the same as any regression approach, is when there is covariance between the environment and a marker, and that also influences the trait. There's nothing we can do about that. That violates our assumptions of causality that we're making in our model. But generally, if there's structure in our data, it seems to be well controlled. When we apply this to real traits, so this is some initial results we have, the proportion of SNP heritability across the different variance components. So I have 5 mixture groups here. One is 0, one is 1% of the SNP heritability, one is 0.1%, one is 0.01%, and one is 0.001%. For height, body mass index, type 2 diabetes, and cardiovascular disease in 460,000 people and 14.5 million markers in the UK Biobank. And we can contrast the traits, and what you see is a big spike here in green, which is for BMI, that the vast majority of the heritability of BMI is attributed to lots of very, very, very small effects. Right? Everything else looks pretty much the same. Right? There's not very much variation here between those two. And I would argue that actually in this scenario, this difference here reflects the phenotypic, what we've called BMI, which reflects a variety, it's a human construct, right? It's your weight divided by your height squared or something like that. And it reflects many different underlying processes, and there's many different ways of having the same BMI. So perhaps the phenotypic definition is actually what's driving this here. When we break this variance up across 250 kilobase blocks of the genome, and we ask, within one block, is there a marker that's always in the model? Right? So we have a window posterior probability of association. And we take blocks that have a marker in the model at least 95% of the time across 10 different chains. Okay? So this, for example, is a window where there's a marker in the model at least 95% of the time, and the window explains 1% of the SNP heritability, right? There aren't any, okay? As we move down, most of them are very small, and there are many thousands of them for every single trait. So if we take all the markers that we have, we block them into groups, we say, okay, for this window, is it in the model at least 95% of the time? Yes, okay, how much variation does it explain? And we group that in this plot. And this highlights actually the complexity of the problem of studying the genetic basis of disease, because, yep.
Wouldn't you expect if you have effects of large size that they create larger LD blocks?
The effects of larger size create larger LD blocks. They don't stay in the model. They tend to fritter between these two groups, which is kind of what's happening here. We can discuss this more at the end of the talk. Anyway, maybe it won't surprise anybody to learn that there are many thousand independent regions of the genome that influence these traits, but it just starts to give us more of a prospect of what the distribution is that we're looking at, highlights for future studies. If most of the BMI variance is sitting here, we've identified what's here in genome-wide association studies. The sample sizes needed to detect things like this of this size are over 2.5 million people before we can get at anything like this on this kinds of scale. Okay. And obviously, running our model, we can predict into the Estonian Genome Center data. And compared to previous methods, this is an approximation of something similar to ours. It's a Bayes C approximation, again using LD to put the data back, to put the LD back in, blah, blah, blah. we can actually improve using the individual level data in what we can predict from traits. So using the individual data seems to provide us with some benefits. In the last just 2 minutes, I just want to introduce a concept about can we extend this model to do some more. And what we seem to avoid a lot is any form of time to event analysis. So we have a lot of phenotypic information about the age at which somebody gets a particular diagnosis. And the age at which they get the particular diagnosis, or the age at which they die, can actually tell us a lot about the genetic basis of the trait, right? Because it can reflect the severity of the symptoms. There might be differences between individuals depending upon the age at which they get the disease. And the problem with time to event data in general is it's left truncated, it's right-censored, and the data is not normal, okay? As I'll show you in a minute. So if I have a study, let's say this. I have a study between these decades here from 1980 to 2000. And 3 people of a particular genotype never entered the study at all because they died before the study began. Some people continued living at the end of the study, and they're obviously censored here, but we have observations for these individuals in the middle. So it's about accounting for data structure of this particular type. And current methods are very ad hoc in the literature, and obviously there's Cox-Lasso, which we'll compare to in a minute, that requires some cross-validation and just really can't handle these types of sample size. And it often leads to poor precision or big bias in the effect size estimates, and there's lots of possible applications of this if we can make it work. So we have a model, and I'm not going through the mathematical detail because I've run out of time, but we have a Bayesian model that assumes the time to event data has a Y-ball distribution, and we use Gibbs sampling combined with adaptive rejection sampling and variable selection in a model of this format to give us estimates of the genetic basis of time to event phenotypes. And we can compare the correlation of, in a simulation study of 5,000 uncensored individuals. Here, this is 5,000 uncensored individuals, and then we add this number of censored individuals to those 5,000 uncensored individuals. And this is the correlation between the true genetic values of those individuals and what we predict from our model to be in an independent set of simulated data. Our model, what we call Bayes W, is up at the top here in green. Cox-Lasso is down here in yellow. And then what the previous studies have done when they've attempted to look at human lifespan is they've tried to take martingale residuals from a lifespan analysis and then treat them as normal. And when we do that either in a standard Bayes R framework, as I've just presented, or least squares estimates, we get these two here. And if you did complete cases, which is also what's been done, you just take the samples for which you have observations and discard everything else, you're going to end up looking like this black dot here. So we always generally outperform Cox-Lasso in every simulation that we've done, and we can account for the right structure of the data. Not only that, but I think one interesting thing that is worth highlighting is that because we have a posterior distribution of effects, we end up with a posterior distribution of a genetic predictor. So we get prediction error for each individual. So not only do we get an estimate of how you lie on that distribution, but your distribution will lie between here and here. And that's been greatly overlooked in the field, that we get a distribution, our predictions have error in them, and that should be transferred from the estimation stage to the prediction stage. And just an example, really rough one, running 100,000 women in the UK biobank of 5 million SNPs, we can look at time to menarche, and we pick up lots of different markers here that are all associated with things that make some sense. And so just to finish, I hope I've shown that large-scale modeling of joint effect sizes has been previously impossible. We've tried to tackle this problem in order to better understand the genetic basis of common complex disease. And I've just highlighted some ongoing work about a flexible model that considers truncation and censoring. And we hope that by doing this, we can better understand why people develop symptoms early. So thanks very much for your attention and time. And I just want to finish by thanking my group, my 2 postdocs, Daniel Treobanis and Thanasis Kousalanis, Marianne and Sven, my PhD students, funding from the Swiss National Science Foundation, the Australian Research Council, and the University of Lausanne. These data sources, and thanks to the participants in those studies, I have a wonderful set of collaborators and mentors to which I'm very, very grateful, and thanks again.